Okay, everybody, this is our AP biology lecture on macromolecules, specifically proteins. So when we talk about proteins, we're really concentrating on some more complex machinery of the cell. So these are biological substances that can alter or respond to other chemical messages, and sometimes even physical or energy-based things as well. These are made of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur, which makes these really interesting bonds that help proteins fold in particular ways. There is no general formula for proteins because they are so complex, but there is a certain part of the molecule that's the same for every protein. So we'll look at that. So they're tools. They do a big list of jobs we're going to look at here in a second. They contain pretty much all the major elements that we talk about in life. Sulfur is kind of the unique one. Okay, so what do proteins do? They give organisms structure. So in humans, that would be things like tendons, ligaments, collagen. A lot of membranes in your body are going to be made out of proteins. So we're going to see lots of structural components of organisms made out of proteins. They make up enzymes, which we're going to do a whole section on enzymes. You guys remember what those are? They kind of lower the activation energy for chemical reactions. They perform very specific functions when it comes to taking molecules apart or putting them back together or altering them. Antibodies. I just read an article that there is an antibody treatment for COVID that's really successful in mice and hamsters. It both prevents it and it treats it if you have it. So we need to get that to human trials as quick as we safely can, because that would really turn this whole thing around, and I'm ready for that to happen. Transport. There's a lot of transport proteins on a cellular level. So hey, let's move this in, let's move this out, and we want to move very specific things in and out. And so we will set up both passive and active transport. On an organism level, you've got hemoglobin, which is a protein that oxygen binds to in your blood. So transport on both the microcellular level and the macroorganism level, okay? Movement. So muscles are a complex set of proteins called actin and myosin, and there's other proteins involved, but those are the main ones. And you throw some ATP in there and you get movement on a sort of molecular level that translates out to, us walking, talking, swallowing, our hearts beating, things like that. Movement can also take place on the cellular level with the cilia or flagella. So you think of like a sperm cell moving. That's not necessarily muscle tissue, but it is protein-based. Receptors. So you have different chemical sort of keyholes on your cells. And when these receptors are hit by different signals like hormones, cells will change their function and behavior. Large organisms are going to need those to maintain homeostasis, respond to the environment. Proteins also make up different hormones, and we saw, don't confuse these. So some hormones are steroids, but not all of them are, okay? So you look at things like, usually you think of these as like the reproductive hormones, but there's also hormones like thyroid stimulating hormone, which controls sort of like, it's like a thermostat for metabolism, and it's going to basically your pituitary gland regulates your thyroid function. So there's all sorts of these messages in large organisms that, again, homeostasis. Okay. Don't have to write down too much on this one. Proteins are what biochemists call polypeptide chains. And those chains are made out of, those chains are made out of amino acids. And we call the bonds between those amino acids peptide bonds. So if you remember, I took that toy that kind of is beads and I hooked them all together. And so what are these individual units called here? They're monomers, and the whole thing is a polymer. And when you make the bonds between monomers, these peptide bonds are still, water out is dehydration, and cutting these, so the dehydration makes the bond. If you want to cut the bond, that's hydrolysis, and it takes water going in. So I'll draw a water droplet there. So these are still hydrolysis and dehydration, okay, even though they're peptide bonds. They're a little different than the bonds between carbohydrates, but they're still hydrolysis and dehydration. Okay, so amino acids have a really, the part of the amino acid I want to talk about right now is the same on every amino acid. This is sort of like the adapter that goes in. So if you think of it like, there's all kinds of different USB devices. There's phone chargers, there's thumb drives, there's cameras, there's mice and keyboards. This is the part that is the USB adapter. The stuff that's hooked onto this is called the functional group. And just like a USB device for a computer, these are all going to be different based on their function. Okay? So on one side of the carbon, you're going to have this carboxyl group, COOH. And I drew it up there on the board. I'll draw it here for you guys who are watching the video. This is arranged like this. You have a carbon double bonded to an oxygen, and then it's also got a single bond to an oxygen over here, and that's going to have a hydrogen on it. So I guess an alcohol or a hydroxyl. So on this side of the carbon, there'll be a second carbon. So what I just drew here is this part right here, okay? On the other side of it, so on this side, there'll be an amine group. And an amine group kind of has that ammonia base. It'll be an N with two H's on it. And I drew it on a separate side for a reason, because they kind of form up to make this amino acid backbone. There's also a hydrogen on it. So let's put it all together. You have one carbon in the middle of the amino acid, and you're going to have a second carbon over here, and that's going to be your carboxyl side, your double bonded O and your OH, okay? So that carbon has 4 bonds, okay? 1, 2, 3, 4. On this side, you're going to have the amine. Hold on, my mouse got a little out of control there. You're going to have the amine group. So with your N, my bonds aren't very straight, so be forgiving. And your 2 hydrogens, okay? Now, this hydrogen right here goes right here. So add a hydrogen right here. And that's sort of what makes the USB plug-in on each end, okay? So we'll talk about that more in a second. Below this, I guess chemically it doesn't matter, but on the picture, draw an R inside that circle. and that is your functional group. So all amino acids have this part here in common. They change on this functional group. And that could be as simple as a single hydrogen to really complex ring structures with totally different chemical configurations. So you have a huge diversity of chemical behaviors and properties in individual amino acids. So why would you need all these different functions? We're going to talk about that. I want you to think about why is there so much diversity in amino acids? Sugars that make up carbs are very sort of regular, and they have the same structure over and over and over again. But this is a lot different. All right. So I did draw the R group on my picture that I just erased for you people at home. It was that thing in the bottom under the central carbon. All right. I have a picture of this that's way better than my drawing. Two pictures, actually. On top, over here, I've got sort of this three-dimensional model that kind of shows this wedge means it's coming out of the page, and this one's going into the page. And then down below, got to point my finger the right way. Down below, you've got a little bit more basic two-dimensional drawing that kind of shows you what I just drew. And these will hook up with each other just like sugars do. You'll get an OH off of one side and you'll get an H off the other. And we'll always see these go nitrogen to oxygen, nitrogen to oxygen, nitrogen to oxygen. And remember, this is your functional group. So this is sort of what determines the chemical properties of that particular amino acid. All right, so these R groups. That's what I'm sort of leading up to right now. The R groups look at all this. You don't have to write this down. I don't care that you know that there are 9, 6, 2, or 3. I just want you to know that there are some that are nonpolar, some that are polar, and then you have some that are acidic and some that are basic. Why so much? Why do we need all these different chemical properties? Okay, here's why. Amino acids do all the biochemical work of every biological process on the planet. So this is kind of neat. All eukaryotes have the same 20 amino acids. Okay, so these are just rearranged in different forms. So it doesn't really matter if you're a pine tree, an ant, or a human. You're going to use the same basic 20 building blocks to get every job that you need done. So this can be kind of really cool when we start thinking about biotechnology, because if a plant uses the same amino acids as you do, we can genetically engineer things to make biological products that they weren't necessarily doing before they're genetically engineered. And we'll talk about an example of that here in a second. Don't have to write any of this down. You'll never have to memorize the shapes of these in this class. But look at the thing that's the same on all of them. The stuff outside of the white box is that sort of amine group on this side and that carboxyl group on this side. The thing that changes is this functional group. These are all nonpolar. You can kind of see some of them have carbon rings. So if you needed to have interactions with that required a nonpolar chemical process, these will be involved. Some of them are polar. So if you wanted to attract polar molecules or fold the enzyme up in certain ways, these would be the ones that you would use. One thing I want you to just notice on this set right here is that we've got sulfur on this one. So we'll talk about what those do. Yes, question? Okay, that's a really good question. All right, let's go back and look at your nonpolars. These are these saturated carbon molecules with hydrogen on them. Those are the same thing that are in your lipids that are nonpolar. These ones, let's go forward, just as an example. So a lot of these are going to have either they're going to be either electropositive or electronegative. So if you think about those OH groups that are in water, remember that OH side, those are going to have some pool on them. So this would have a negative pool. The sulfur ones are going to be they're going to pool together. Okay, so these will make, the polar ones are going to sort of make chemically weak hydrogen bonds for the most part. And it's just a matter of, and I'm getting a little bit out of my chemistry element here. It's just a matter of how electronegative these molecules set up as. So the electron in this OH right here spends more time around either the O or the H, and that makes it polar. These electrically charged ones, these are basically ionic, okay? So the acidic ones here have negative charges, so these are looking for something positive to bond with. And the basic ones have positive charges, so they'll be looking for something negative to bond with. And you can kind of see those are setting up right there. All right, R groups are what we just looked at, all those different R groups. Don't have to memorize any of them. One thing I want to point out to you, if you can see it, is all these amino acids have nicknames that are sort of like 3-letter abbreviations. So you'll start seeing in biology, you'll start seeing like these circles, and they'll have like ASP, and that stands for aspartic acid. I didn't draw that very good. This one will have GL, and they'll make the chain of amino acids. If you remember, it takes 3 bases in your DNA or RNA to signal which amino acid. We'll get to that stuff later, but every 3 bases is a code for one of these particular amino acids. And the code a lot of them have multiple ways to do the code, but that's not where we're at right now. Okay, so R groups, some of these are going to have the stop code in a protein or the start code. That should have been stop or start. I'm not sure why it's the S there. So some of these are going to have that, and some of these are also going to basically, the properties of the R group determine the properties of the protein. And this is like building small Legos that build bigger Legos, that build bigger Legos, that build some final Lego factory. Okay, so they just kind of keep folding and folding and folding. So what do these chains do? Okay, I just kind of want to remind you, okay? And when I say AA in this lecture, that's sort of like my shorthand for amino acids. We already talked about this, but the chains are made by dehydration. And so you're forming a bond between the carboxyl side and the amino side. One of the differences here between carbs and proteins is the sugars can go together in different ways. And we looked at like how putting them together in different ways determine their structure. These only go together in one way. The carboxyl has to join the amino. You cannot join a carboxyl or amino. So they're kind of like extension cords, okay? You have the outlet side, and then you have the plug side. If you were going to put a bunch of extension cords together, you can't put 2 plugs together. You can't put 2 outlets together. All right. So that backbone that we're looking at, here's how it sets up. Okay, right below my picture, you'll see the OH group on the carboxyl side, and you'll see one of the hydrogens from the amine side. And you can hear that's amino acids. And so it'll chop one of these off, this OH group here, and you get dehydration again. And we call that a peptide bond. In sugars, it was a glycosidic linkage. Not really concerned that you have those memorized, but on the end of every amino acid, no matter if it's 3 units long or if it's millions long, you'll have an amino end and a carboxyl end. So if this was like 3 extension cords, you'll still have like a plug on one end and an outlet on the other. Okay, so I'm just trying to, when I say the plug and outlet, I just want you to understand they only go together one way. Okay, they only go together one way. You don't have extension cords with like 2 plugs or 2 outlets on them. Okay. Proteins have a couple different levels that they fold up into. And they start as almost like a necklace with beads or a strip. And then they become, this is very one-dimensional. I mean, it's not truly one-dimensional, but it's just more like a string, okay? Or a strip. Then they can make 2 types of structures. So imagine you take sheets of paper and you pleat it. If you don't remember what pleat means, that's like where a fold was. So if you fold we talk about folding paper, hamburger, and hot dog. So the fold line would be a pleat. And you can kind of see these make that zigzag and almost like a springy look or a pasta look or something. Those are called beta pleated sheets. Then they make these helix shapes, and they're called an alpha helix. So this is the primary structure, the strip. The secondary structure is the either beta or alpha. Then they make these things called tertiary structures. Now, if you look in there very closely, you've got pleats, you've got helix, and then you have some sort of strip spots that are still primary. So all 3 of these make this tertiary type structure. Now, it's not called tertiary because there's 3. It's called tertiary because it's the first, second, and third level. Multiple tertiaries, so these little jelly bean looking things that are full of amino acids, come together to make these quaternary structures. And these are sort of the complete final protein. So proteins have multiple steps they need to get to be built into a functional unit. If there was some mutation or environmental factor that stopped one of these from going into the next form, that protein would be non-functional in that organism. So that could be maybe we put some sort of toxin in that stops that. Maybe there's a mutation that causes the shape of this to be wrong. And we'll look at some examples of that. So that organizes the chain into primary, secondary, tertiary, and quaternary. So let me ask you this. What's below primary? Sets of what? Sets of amino acids, okay? All right. There's a primary. That's just a long, long chain. And you can, if you look very closely, there's 3 letters on each one. That's those sort of 3-letter abbreviations or sort of like shortened names of each of the 20 amino acids. All right. So the primary is the sequence. Now this has to be in a very particular order. These aren't just random. DNA and RNA tell your cells what order to make these in. And that order is super important. A mutation would take one of these out, or it would frame shift it, which would screw the whole thing up, and you would have probably a non-functional protein. So with 20 amino acids, and you make thousands of different combinations, probably Actually, it's probably in the high millions or even higher. When you put thousands of these together, you can have these really, really, you have a lot of variation in what you can get done. One thing just to pay attention, it has an amino end and it still has a carboxyl end. All right, your secondary, it's going into a 3D structure, okay? And this is mainly with different bonds, hydrogen bonds, making those pleated sheets or those alpha helix. Well, I showed you what those look like. A helix is a spiral, a pleated sheet, kind of that folded paper. Okay, so spiral or folded ribbon of amino acids. Okay, so here's what they look like close up. I did draw them for you. There's your spiral, there's your zigzag folds in your beta sheets. When you look at proteins, try to notice that these are out there. There's a spiral right there. That's your alpha helix. There's a folded sheet right here. That's a pleated sheet. Now these things will twist and bend quite a bit when this thing's doing its work. Tertiary. This is when instead of that hydrogen bonding, we have all these different classifications of bonding. The best way to show you this is in a picture, but you've got hydrophobic, ionic, and these. This is why sulfurs. I kind of keep bringing up sulfurs. Sulfurs make these covalent bonds that kind of fold back the protein and connect it together really strong. So I'm never going to ask you what these bonds are or to I'm not going to ask you to identify these, but I just want you to know, in a basic science class, we might just go, and amino acids fold into proteins. Well, why? How? What makes them fold? So and sometimes in lower level, we simplify stuff to the point where it's just like, this happens. Well, it doesn't just happen randomly, and it doesn't happen it doesn't have muscles or its own, mind trying to fold up. We have all these different tools to fold proteins because they do so many different chemical jobs that they have to have all this versatility. So the hydrophobic stuff might be pulling away from water into a cluster. These hydrogen bonds here, those are going to be weaker. The disulfides are going to be your strongest, and your ionic bonds will probably be second strongest. So all these different ways of folding don't just happen. They're driven by chemistry. All right. Quaternary structures are sort of the final form. And look, multiple blocks of tertiaries will come together. This is collagen, which makes fibers in your skin and in your tendons. It's very slow to heal in your body because it doesn't have tendons and things like that don't have a good blood supply. So as you age, your collagen gets worn out and your skin gets droopy. This is like a four-leaf clover. You can kind of see the four sections here. Each of these four leaves of the clover has an iron molecule in the middle of the protein. Each one of these leaves carries an oxygen molecule, an O2 molecule, in your blood. So red blood cells don't have a nucleus. They don't have ribosomes. They're just bags of this protein. And that's going to deliver all the oxygen you need to your body. If you don't have enough iron, you can't make this protein. And they call that anemia. So low iron people. It's not really the iron you need. It's the iron to make the hemoglobin. All right, so the final form, this fourth form, is when these guys unite the tertiaries. What makes these different? This protein is functional. All those tertiary levels, I think there's a few that are functional at that third level, but they have to turn into these big molecular machines. So these are little biochemistry machines that get all kinds of different jobs done. All right. Structure is super, super important. One little change in one of those amino acids pretty much either makes the protein non-functional or very, very limited in function. Now remember, you have 23 pairs of chromosomes. So you might have a functional copy of the gene on one chromosome. Say this one's from dad and mom's the carrier. You might have a non-functional one on this side. So you might make 50-50. You might be like, well, half my proteins are right and half are wrong. This is why if both, for a lot of genetic diseases, it's not really traits that you might see, okay? It's going to be stuff on the protein level. And, well, I guess you do see stuff on the protein level sometimes too, like eye color. But when you see one little change, we go from normal red blood cells, which are kind of like Cheerio looking, to sickle cell anemia just from one change. So this thing basically can't form a proper structure because this one amino acid change. All right. Here's some sort of technical drawings on why it doesn't fold up right. Let me just go through this really quick. You have these subunits of proteins, and they keep sort of making these different structures. And in sickle cell, that doesn't form right. And you get this banana shaped, or a sickle is a curved knife, versus the Cheerio shape or the trampoline with somebody sitting in the middle. Okay, so you can put proteins together and you can also take them apart. Let's pause here and we'll come back and talk about denaturing proteins.